So my lab focuses on a different kind of immune receptor, an immune receptor that is not part of a currently approved cell therapy, the T-cell receptor, as opposed to a chimeric antigen receptor, or CAR. And the reason we're so interested in the TCR is that it allows an immune cell to recognize antigens or targets that are located within a cell, either in the cytoplasm or nucleus. And the reason that's important is that allows a cell to perceive maybe 90% more targets than is possible with a chimeric antigen receptor. And many of these targets that are inside of a cell, a cancer cell, are either tumor selective or even tumor specific, reducing the risk of unfortunate side effects. Yeah, so the magic trick of the TCR, of this immune receptor that allows one cell to look inside of another cell and ask the question, are the proteins there normal or abnormal, is really a magic trick, meaning that there is more than meets the eye. So the magic trick is that all proteins, including abnormal proteins, are continuously recycled within a cancer cell through a process called proteasomal degradation into smaller peptide fragments. And those peptides are loaded into a molecular basket called HLA, which is shuttled to a tumor cell surface. So that's the magic trick of how a protein or pieces of a protein located on the inside of a cancer cell can be displayed to the external universe for a T-cell receptor to see. So our focus in the laboratory is to try to identify targets that overcome many of the limitations of other TCR therapies. So in particular, we're looking for targets that are shared among groups of patients, don't require an individualized approach, target proteins that are clonally or homogenously expressed, and targets that are very, very difficult for a cancer cell to lose and still be a lethal metastatic disease. So the approach we've taken is to immunologically target recurrently mutated proteins called driver proteins, which when activated or mutated inside of a cancer cell, function basically like a stuck-on accelerator. So in my talk, I talked about a family of shared neoantigens, or what we've termed public neoantigens, derived from a protein called NRAS, which is very commonly mutated, especially in patients with difficult-to-treat melanomas, as well as difficult-to-treat colon cancers and cancers of the thyroid.